The overall goal of this procedure is to prepare Drosophila pupae for live cell imaging of dividing thoracic sensory organ progenitor, or SOP, cells during metamorphosis. This is accomplished by first setting up the appropriate genetic cross to obtain progeny pupae for live cell imaging. The second step of the procedure is to collect Drosophila pupae at the appropriate stage to visualize asymmetrically dividing neural progenitor cells. The third step of the procedure is to remove the pupal case in order to mount the pupa for imaging. The final step of the procedure is to mount the pupa between slide and cover slip and proceed to live cell imaging. Ultimately, results can be obtained that show the dynamics of fluorescently tagged proteins during asymmetric cell division and differentiation of wild-type or mutant sensory organ cells through either epifluorescence or confocal microscopy.
The main advantage of this technique over existing methods like immunohistochemistry is that protein dynamics can be followed in real time and over the course of several hours in an intact pupa.
From a cross of the desired GAL4 line and GFP-tagged fusion protein under UAS control, identify pupae that have undergone pupariation within the hour. These pupae will possess characteristic pupal morphology but lack pigmentation. Selected pupae can either be transferred to a fresh file or can be marked on the cross file to remember which ones were white 18 hours later. When the pupae reaches the appropriate stage, place a piece of double-sided tape onto a microscope slide and adhere the pupal case to the slide, ventral side down. Under the dissecting microscope, identify and grasp the edge of the operculum using a pair of forceps and gently lift it off, revealing the head of the immature fly. Being careful not to puncture the body of the fly, gently tear the side of the pupal case and remove it completely to reveal the thorax and anterior portion of the abdomen. Continue to carefully tear the same side towards the posterior and pull the pupal case to the opposite side and stick it to the tape. Now that the abdomen is fully exposed, gently push a soft bristled brush under the head of the pupa and lift it from the rest of the pupal case. Place the pupa onto a microscope slide, dorsal side up. Position the pupa at a 45-degree angle so that the cover slip will touch the thoracic SOP cells region first. Make an 18 by 18 mm square frame out of Whatman filter paper by cutting a 10 by 10 mm window in the center. Immerse the frame in water until saturated and then place around the pupa. Use a 5 cc syringe to apply a uniform layer of silicone grease around the frame such that the layer's thickness is only slightly greater than the pupal diameter. Pipette approximately 1 microliter of water on the center of a 22 by 22 mm cover slip and position the cover slip such that the droplet touches the pupal notum. Press gently to form a complete seal and flat surface against the pupa. Now that the live pupa has been mounted, it can be imaged using confocal microscopy. By combining the dissection and mounting techniques with live imaging, one can visualize the accumulation of actin GFP fusion protein at the cleavage furrow of a mitotic SOP cell over time. Here, the distribution of partner of NUM RFP and Rab5 GFP can be compared across two SOP daughter cells. Note that while partner of NUM RFP is asymmetrically distributed, Rab5 GFP is distributed equally in both cells. At late pupal stages, differentiated external sensory organs can be visualized. Mechanosensory sockets and hairs exhibit autofluorescence and are shown here overlapping with LGL-GFP expressed in a subset of SOP cells using the MARChem system.
Once mastered, this technique can be done in less than 10 minutes if it is performed properly.
Following this procedure, other methods like immunohistochemistry can be performed in order to answer additional questions like identifying cell fates of sensory organ cells with specific antibody markers or colocalization study of GFP fusion proteins with endogenous proteins.